are harvested from different niches in an adult person's different organs. Almost all our organs have a reservoir of stem cells. So we can remove stem cells from organs of an adult person. Typically the most common sources, the most abundant sources are either fat or bone marrow. So these are the two sources, two type of stem cells you will encounter in literature, being most commonly used for therapeutic purposes. There is no ethical dilemma. Of course, you're taking it from a person, and adult person who has already consented that their tissue be removed for preparation of stem cells. However, they have a limited ability to make more copies of themselves and also differentiate into different types of tissue. That was our initial goal, that we want a cell that can make as many copies as many we like, and can differentiate into any type of cell specific to that particular organ. So the ability here is slightly limited. There's another type of cell you will also encounter in literature that is induced pluripotent stem cell. So normally our cells in our body that have already chosen a profession, जैसे मैंने बात की थी कि जो हमारे adult stem cells हैं, heart के cells हैं, उन्होंने एक PhD की हुई है. इसी तरह जितनी सेल स्पेशलाइज्ड होता जाता है, उतनी ही उसकी ability to make more copies of itself and differentiate into a different type of cells जो है कम होनी शुरू हो जाती है. Ability to specialization of a cell is inversely proportional to its therapeutic potential. So what we can do, what basically this technique does, is it takes a specialized cell, generally fibroblast, and then we can artificially trick it into thinking that it is a stem cell of an embryonic level stem cell. It can make many copies of itself and can also differentiate into different types of cells. So this is done by four different factors. I'm not going to go into them any detail, but however, OCT4 and SOX2 are important for cell renewal and also making a differentiated cell into an undifferentiated cell. आप इस तरह से समझें कि एक PhD पास सेल को हम पीछे घुमा के कच्ची पास का बनाते हैं. So these two, OCT4 and SOX2, they play an important role in that. C-MYC regulation, proliferation, differentiation, apoptosis, and somatic reprogramming. That is the function of the C-MYC. It's a very important protein that can do this. And also, GIF4, it also helps in cell proliferation. So, a normal adult human cell differentiated can be converted back into a stem cell with the same potency, same effectiveness as an embryonic stem cell. Next slide. Umbilical cord mesenchymal stem cells. This is a happy medium. So they are adult stem cells. They come in the adult stem cell category. These cells are prepared for umbilical cord, and the beauty here is that these cells have a much greater ability, regenerative ability, as compared to the stem cells that are obtained from fat or bone marrow. They can make many copies of themselves. And also they have a much greater ability to differentiate into different types of tissue. So for a spinal cord injury patient, these will be more better suited, or neurological diseases, this type of cell will be far better suited because of its regenerative potential, which I defined as the ability to make more copies of itself and also differentiate into different type of tissue specific cells. So the beauty here is that what makes them more important therapeutic tool is that they do not express MHC molecules on their surface, major histocompatibility of human nucleoside antigens on their surface, so they are primarily ignored by our immune system. So they can survive, they can be a very important therapeutic tool. SVF, that is what we are going to make today, inshallah, in the lab. Stromal vascular fraction. Okay. So this is not a pure population of stem cells. It has a mixed population. It has mesenchymal stem cells, it has epithelial progenitor cells, it has endothelial cells, fibroblasts, and also different pericytes, for example, mentioning here. So it is a mixed type of cell population. इसका फायदा क्या है? MSC जो है इसके अंदर जो mesenchymal stem cells हैं वो roughly 5 से 6% हैं, जिनको हम stem cells कहते हैं traditionally. लेकिन इसके अंदर और भी बहुत सारे किस्म के cells हैं. लेकिन in many diseases, when you want tissue to regenerate, you need some other activities also. For example, you need angiogenic activity. So epithelial progenitor cells and pericytes, they play a very important role in formation of blood vessels. So if you are going for a systemic approach, किसी की किडनी खराब भी है और एक ऑर्गन किसी का अगर जिस्म में खराब हो, ultimately it will result in compromise on other organs. मैं इसकी मिसाल इस तरह से देता हूँ, गाड़ी का अगर एक पुर्जा खराब हो गया, छोटा सा कार्बोरेटर खराब हो गया, तो अगर आप उसको रिप्लेस नहीं करते, ठीक नहीं करते, अल्टीमेटली इट विल रिजल्ट इन ओवरऑल गाड़ी के बाकी के जो पुर्जे हैं उसके ऊपर भी असरअंदाज होगा इसका एक पुर्जे का खराब होगा. सो व्हेन यू आर गोइंग फॉर सिस्टेमिक अप्रोच, these cells are better in many ways than pure mesenchymal stem cells. अगली करते हैं bone marrow mesenchymal stem cells or mononuclear cells. Bone marrow is extracted from iliac crest through biopsy, and these bone marrow, it is processed in a way that we get a fraction at the centrifuge, a fraction that has mononuclear cells. Of course, it is again going to be a mixed population of cells. These cells can also be used for therapeutic purposes. Actually, पहले adipose tissue पे जाने से पहले this was primarily the source of stem cells. लेकिन it turned out that adipose tissue has far greater stem cells than bone marrow, so people slowly switched to SVF rather than bone marrow stem cells. So there are certain things that I would like to highlight today. Hopefully, inshallah, I would like to hear that many of you have started with their own stem cell facility and they are treating patients successfully. उसमें कुछ एक दो चीजें मैं आपको बताना चाहूँगा कि how I deal with patients. सबसे पहले तो patients जो हैं उनको ये देखना चाहिए कि जिस बंदे के पास जा रहे हैं for stem cell therapy, whether that person has the right education and qualification to do stem cell therapeutics or not. That's number one. Training उसकी हुई है या नहीं, और कहाँ से हुई है और कितनी ट्रेनिंग हुई है. Technical know-how of that person and जो है honesty is also very important क्योंकि you have to अब वो टाइम गुजर गया है कि डॉक्टर पेशेंट को बता देता है कि आप बस ये कर लें और ये हो जाए. Now our patients are very well informed, तो उनके साथ we have to treat them with respect and we have to give them the right information. आप उनको लिटरेचर दें, उनको बताएंगे ये जो थेरेपी है इसका ये फायदे हैं, ये प्रोबेबिलिटीज हैं, and then let patient decide. You have given them the option, you have given them all the information, you have given them the literature, and then you have to respect whatever decision they make. अगली स्लाइड करें, मैं आपको एक पेपर दिखाना चाह रहा हूँ. वो जो पेपर है वो ऑस्टियोआर्थराइटिस के पेशेंट्स फॉर एग्जांपल अगर आते हैं तो इन मेनी केसेस जहाँ पे अवेलेबल होता है आई गिव देम लिटरेचर टू टेक होम, लुक एट देम, दे कैन डू कंसल्टेशन विद अदर पीपल, देयर फैमिली मेंबर्स एंड देन मेक द डिसीजन. इसको अगर आप नीचे देखें, दिस इज अ मेटा एनालिसिस. इसके अंदर मेरा ख्याल है 30-40 डिफरेंट स्टडीज stem cell therapy for osteoarthritis की नीचे टेबल्स आए हैं, टेबल्स हैं और उनका आउटकम इसके अंदर क्या है. ले जाओ और टेबल्स आए हैं.